Hi, my name's Rob Patterson, and I'm going to try to teach you protein synthesis. Now the first process is transcription. Here I have made a DNA model. DNA has things called the nucleotide bases, which are all the colors here. Now there are 5 types of nucleotide bases. There is adenine, cytosine, guanine, thymine, and uracil. Now here I have marked a letter for the start of each of them and also color coordinated them. So orange is adenine, cytosine is red, guanine is green, thymine is blue, and uracil is yellow, but it will not be shown until the mRNA is being made. So what happens here is A's and T's always pair up and G's and C's pair up. So orange and blue and green and red. Now what happens is the helicase comes in and splits up the DNA, and you get your leading strand and your lagging strand. Now to make the mRNA, they use the leading strand, and they use the leading strand to copy off of for making the mRNA. Now the mRNA is pretty much just a copy of the lagging strand, but thymine is replaced with the uracil. So it's pretty much the exact same, but thymine is now uracil and it's yellow instead of blue. So there we are. So the leading strand is used for copying to make the mRNA. Process 2 is translation. So here is my nucleus and my mRNA, which are not to scale whatsoever. So the mRNA leaves the nucleus, goes, and a ribosome forms around the mRNA, and along the mRNA are things called start and stop codons. There's one stop. They're just variations of nucleotides. This is only one start codon, which is AUG, and there are 3 stop codons, which is UAA, UAG, and the one we use, which is UGA. So what happens is the tRNAs go along the mRNA until it finds its match, which is right here. AU, AU, and GC matches up, and it leaves an amino acid. And then another tRNA comes along till it finds its match, which is right there, and it leaves another amino acid. They bond together, and then this process keeps going on, matching up and leaving the amino acids until it hits the stop codon, and that chain of amino acids are called polypeptide chains, which leave the ribosome to form proteins. Types of mutation. I'm going to give an example of deletion and insertion mutation. Insertion mutation is when for example, cytosine is added in between the A and the U of the start codon. This would affect the mRNA because as the tRNA goes along reading the mRNA, it wouldn't find its start. So the polypeptide chain would never get started. An example of deletion mutation would be to lose the G at the stop codon. This would affect it because as the tRNAs go along to finish the polypeptide chain, it wouldn't be able to stop the polypeptide chain, so it would keep going.